Hello everybody, this is Aloysius Guy in a new tutorial, and today's topic is about chromosomes. So we're going to understand what are chromosomes, what's their structure, what is their function, and before we start, please don't forget to check the time codes in the description. Let's start. So we will start with what type of chromosomes we have in the human cell. So in the human cell, we have two different types of chromosomes. So the first type of chromosomes are called autosomal chromosomes. And the main characteristics of autosomal chromosomes is that they contain genes that are involved in determining individuals' hair color, eye color, individuals' height, also skin color. So all these general characteristics are determined by genes found in these autosomal chromosomes. And in humans, there are 22 pairs of autosomal chromosomes. Autosomal chromosomes, they are found both in males and females, and they are inherited from the parents equally. And most importantly, that autosomal chromosomes do not contain any genes that determines individual sex. As we can see here, that each pair is made up of two chromosomes. The one chromosome is inherited from the mother, and the second chromosome is inherited from the father. And that's true for all chromosomes. The second type of chromosomes that is found in a human cell is the sex chromosome. So sex chromosomes, they have also specific characteristics. And the first thing is that they carry genes that determine the individual sex. So in the human, there are two types of sex chromosomes. There is an X chromosome and a Y chromosome. And the main difference is that the X chromosome would carry genes that are involved in both males and females characteristics. However, the Y chromosomes, they only carry genes that are specific to males. And that's why when we look at the pairs here, which is also similar to autosomal chromosomes, they are found in pairs, okay? And each pair contains two chromosomes, one from the mother and one from the father. And since Y chromosome only contains male specific genes, that's why a male individual would have X and Y, a female individual would have X and X, okay? So within the cells of the human, there are somatic cells which contains all these chromosomes. So they have 46 chromosomes or 23 pairs. Okay, so 22 pairs are autosomal chromosomes, and the last pair is the sex chromosome. It's either XY or XX. The exception for that is what we call is the haploid cells or the gametes, which are sperm and eggs, and these cells will have only 23 chromosomes. And the reason for that is that because half of these chromosomes will be splitted and moved into the sperm, and the same thing happens in females, so they have only 23 chromosomes. We will cover in more details how the haploid cells are formed and what happens to chromosomes during meiosis and mitosis in a future video. So all we explained so far is about the number of the chromosomes and the types. So now we're going to get more into the structure of the chromosomes. So as we can see, they are all thread-like structures, and they are all found within the nucleus. Okay, and when it comes to the content of these chromosomes, each chromosome, it's basically a single continuous DNA molecule that is highly compacted to form this thread-like at the end. Okay, and having chromosomes, it's very efficient, is very important for having an efficient DNA storage, also for transmitting the genetic information to the next generation. Also, they are important for a stage called segregation, which happens during cell division, and we will cover that hopefully in the future in a future video. So now we need to understand why chromosomes allow efficient storage for DNA. So as we know, within the human cell, we have a nucleus, and within the nucleus, there is the DNA or the genetic material. So If this DNA within each nucleus, within a single human cell, if that DNA was to be unwrapped, it would form a line that is approximately 2 meters long. So that's only from a single cell. So that DNA is highly squashed within that nucleus in few micrometers wide within the cell. So if we imagine that we took the DNA from all the cells within a single human being, which in every human there is approximately 37 trillion cells, and if we extracted that DNA and then we made it a rope into a line or a rope, that rope will round or wrap the Earth 2 million times. So that's why having chromosomes and developing mechanisms to squash the DNA and compact it is very essential for our very being. Because without chromosomes and without high compaction of DNA, the cell wouldn't be able to fit the DNA within it. So since DNA compaction is very essential to the cell, now we need to understand and to know what are the stages of DNA compaction to become a chromosome at the end. So there are several stages of compacting the double-stranded DNA until it becomes a chromosome. So the first and the simplest conformation of the DNA is having the DNA double helix, and that's the simplest conformation. And then it comes a second, a much higher compaction, which is called nucleosomes. The second stage is called the 30 nanometer chromatin fibers, and then the formation of what is known as the loop domain, which would have these forms, and finally having the chromosome in that form. So this DNA will be compacted through all these stages until it becomes a chromosome. And in this tutorial, we will go through every stage and we will understand how DNA can become chromosomes at the end. So the first level of compaction of DNA is the formation of nucleosomes. So what are nucleosomes and how are they formed? Within the human cell, we have a protein called histone. Histone has 4 different subunits: H2B, H2A, H4, and H3. And within a histone octamer, there are 2 copies of each histone subunit. So there are 2 copies of this, 2 copies of that, and of this. So what happens is that the DNA will start wrapping and twisting around the histone octamer. And within around every histone octamer, there is approximately 1.65 turns of DNA. So the reason the DNA twist around the histone is that the DNA is a negatively charged molecule, and they will start interacting with the positively charged amino acids found in the histone octamer. This will allow the DNA to start twisting around the histone in an anti-clockwise manner, forming what is known as a superhelical structure, or also known as nucleosome. So a nucleosome is the histone octamer protein with the wrapped DNA around it. So these together are called a nucleosome. Okay, and within every nucleosome, the amount of DNA that is twisted is approximately 147 base pairs of DNA. So this process will occur several times, forming a structure resembling beads on a string. So the beads are the histone core and the string is the DNA wrapped around the beads and in between. And every one of these is called a nucleosome. We can see that between nucleosomes there is a space of unwrapped DNA, and this DNA is called the linker DNA. So we have the DNA that is within the nucleosome, and we have a DNA that is called the linker DNA found between nucleosomes. So the second level of compaction after formation of nucleosomes is what is known as a chromatin, or the formation of a chromatin. So what is a chromatin? So as we just explained, that now we have here 2 nucleosomes. Each nucleosome is wrapped around the histone octamer, and each DNA is wrapped around the histone octamer, forming a nucleosome. Once these nucleosomes are formed, a new isoform of histone called histone 1 will interact with nucleosomes. So what happens is that histone 1 will interact with both the linker DNA, which is the DNA found in between of the nucleosomes. So it interacts with the linker DNA and with the exit and entry regions of the nucleosome. Okay, so in total, the core histones with histone 1 with wrapped DNA, they are all called a chromatin. So chromatins have different levels of complexity, and this structure or this form is the simplest form, or the simplest unit of chromatin. So the presence of chromatin histone 1 is very important for the compaction of chromatin and into higher order structures that are more complex, and they are more compacted, and all that is in the journey to form a chromosome at the end. So what happens is that once histone 1 interact with this nucleosome and interact with this nucleosome, now they will facilitate the formation of a new level of compaction, which is called the chromatin fibers, or known as 30 nanometer chromatin fibers. So these chromatin fibers have a very condensed fiber-like structure. They also considered to be a level of organization that is beyond the nucleosome. So if you can see that we started with a double helix DNA, then we became a nucleosome, the nucleosome became a chromatin, and then these chromatins started also folding further with the aid of histone 1 to form these fibers. These fibers can undergo reversible changes, and these changes are important because it allows the interaction of chromatin with surrounding proteins, and also is very important for gene expression. And we will understand later how reversibly compacting these fibers is very important for the access of RNA polymerase and all the components of gene expression. And if you just look at the link that will appear here, and there will be more information about how gene transcription can occur, and how RNA polymerases is very essential, and they need to access this wrapped DNA. So now we get to the last few stages of DNA compression and compaction to form chromosomes. So as we have just mentioned, that these chromatins will form a chromatin fiber, which is known as 30 nanometer chromatin fiber, and the name 30 nanometer comes from the width. So these fibers are 30 nanometer wide. The next compaction stages involves forming loops of these chromatin fibers. So as we can see, this chromatin fiber will start looping, forming these loops. The benefit of having these loops is that they facilitate the interaction between genomic regions that otherwise would have been so far away. So for example, a genomic region on that loop here and a genomic region on that loop here, they would have been too far away. However, looping allowed them to get very close. So that's very important for gene regulation, DNA replication, and also for DNA repair. And then we can see that they're going to be also even further compaction of the chromatin, forming these fibers, until at the end they will form a highly compacted chromosome, or sometimes they are called chromatids. So chromatid is basically an identical copy of a chromosome that is usually occurs during cell division. But as I said before, we will cover that in a future session. And this is the whole process of compaction and compressing a double-stranded helix of DNA until it becomes a chromosome. So, so far we have distinguished and classified chromatin based on their folding and their compression states. However, we can also classify chromatin based on their function. And the first type of chromatin based on the function is called euchromatin. And the main characteristic of euchromatin is that they are less condensed form of chromatin. Also, the DNA is accessible for transcription factor and also for DNA polymerases to interact with and to initiate processes like gene transcription. And if you check the video here, you can get more information about gene transcription. Also, euchromatin, they are considered to be transcriptionally active and accessible to cellular machinery. The second type of chromatin is called the heterochromatin. And heterochromatin, they characterized with being highly condensed form of chromatin, so they are more condensed than euchromatin. They are also considered to be transcriptionally inactive or repressed. And the gene that is usually found within the DNA that is wrapped around the heterochromatin region, they are considered to be genes that are not expressed or genes that are expressed at a very low levels. Okay, having heterochromatin is also very essential and supporting and maintaining chromosome structure and stability. And that's generally how or the two classification of chromatin based on their function. So before we continue with this session, please don't forget to like, comment, subscribe, and also hit the notification button so you can get notified. So now we need to answer a very important question. So when chromosomes, are they visible within the cell? Or is the DNA within our cells always found in the form of chromosomes that is very highly compressed? And that's what we're going to try to answer now. So as we know that each cell go through something called the cell cycle, which is the cycle that ends by having two daughter cells from one parent cell. So it starts here and then go through the division until it produces two daughter cells. So within the interphase, we have this stage here. So this stage is the longest stage of the cell cycle, and the cell stays the longest in that phase. And basically, when the cell is in that phase, we say that the cell is not dividing. Once the cell go into the prophase and metaphase and these phases, now we say that the cell is dividing. So within the interphase, the chromatin within the nucleus will appear as a DNA network. So all this network is basically a chromatin that is a DNA that is wrapped around chromatin and then folded. However, the folding is not highly compressed until chromosomes are visible. So it's going to be a network like that. However, in the other stages, as we can see here from the image, that we can see the structure of chromosomes within these phases. So what happens is that during cell division, the DNA gets even more highly compacted than what the cell usually have in the interphase. So they get highly compacted into chromosomes, and that's very essential and important to facilitate the division and the duplication of the genes and the DNA, and that ensures that every daughter cell would have a complete set of genomic DNA. And to know more information about DNA replication, just click on that video here and you can get more details about DNA replication. So now we need to get into more details about the structure of the chromosome. So as we can see here, this is the complete set of chromosomes found in a human somatic cell. And we can see a very common thing between all chromosomes is that they have this narrow area within each chromosome. So what is this narrow area? So these areas are called the centromeres. Okay, so all human chromosomes have centromeres. These regions are very specialized, as they are very important for facilitating chromosome segregation. Also, they allow the chromosome to be splitted into two different segments, and these two segments are called chromosome arms. So each chromosome would have two arms or two segments, a short arm which is called the P arm and a long arm called the Q arm. These two segments or these two arms will be structurally distinct, and also they will carry different DNA sequences, and therefore they are carrying different genes that are expressing different proteins. And that's true also for the sex chromosomes, as they have centromeres, and also they have the P arm and the Q arm. And in addition to that, we can see that all chromosomes are found in pairs within human cells, the somatic cells. So each of these pairs are called homologous chromosomes. So in a diploid organism like a human's, somatic cells would have all chromosomes in pairs, as we said, and these pairs are called the homologous chromosomes. So also, if you look at the bands that are here on each of these chromosomes, these bands are called locus. So the locus is an area within the chromosome that is occupied by a specific gene. So if you want to know what is a gene and how to know how to distinguish a gene from another DNA sequences, just try to click on the link that will appear here and you will get more information on that. So also, if you look here, so this is the area of the centromeres. So what are the characteristics of a homologous chromosomes? So homologous chromosomes, they carry the same gene at the same loci. So the word loci is just the plural word for locus. So locus is just an area of a single gene. Loci, here we are referring to the different areas within the chromosome that are occupied by genes. Okay, so the first thing, that homologous chromosomes would be carrying the same genes at the same loci. So if this is a gene that is determining the eye color, then this is going to be the other version of the gene, which is called allele, and that is also determining the eye color. Then also they are found in similar sizes, similar shapes, and also the position of the centromere are going to be in the same position. However, homologous chromosomes are not identical chromosomes, and that's again because they might carry genes of different versions, so they might have alleles. We will do a future video on that, explaining what are alleles and how they are, and how a single gene can have different alleles. And that's in general the main structure of homologous chromosomes. Okay, so now we're gonna get introduced to chromosomal abnormalities. So chromosomal abnormalities come in two different forms. And here we're gonna cover now the first form, which is alteration in the number of chromosomes. Of course, there are different conditions with different abnormalities in the number of chromosomes, but here we're gonna just cover two different examples, trisomy and monosomy. So trisomy is a condition in which there is an additional copy of a chromosome. As we now know that within the somatic cell in humans, all chromosomes are found in pairs. But what happens in this condition is that one of the pairs will have an additional copy of chromosome. A very well-known example is the Down syndrome, or it's called trisomy 21. The reason it's called trisomy 21 is that because in chromosome 21, what happens is that they're going to be an extra copy of chromosome 21, as we can see here. So instead of just having a pair of chromosomes, they're going to be 3 chromosomes in that pair. And this will lead to different complications like intellectual disability, characteristic facial features, and also various health complications from just having an extra copy on chromosome 21. The second type is monosomy. So monosomy, in contrast to trisomy, it involves the absence of one of the chromosome pairs. And a known example for that is the Turner syndrome, which affects females, and it involves that within the X chromosome in females, as we know now, that in females they have 2 copies of the X chromosome. However, females with Turner syndrome would have only 1 copy of the X chromosome. This will result in and lead to different complications like short stature, ovary insufficiency, and also some heart defects. So the second type of chromosomal abnormalities involves alterations or changes in the normal structure of the chromosome rather than the number of chromosomes. So here change is going to affect the structure of the chromosome itself. Of course, there are different examples of changes in the structure of chromosomes. However, today we will just focus on translocations and deletions. So translocations, they involve an exchange of a chromosomal segment between non-homologous chromosomes. And one known example of that is known as the Philadelphia chromosome. And Philadelphia chromosome is associated with having myeloid leukemia. So what happens basically is that within chromosome 9, this segment will break off from chromosome 9 and then attach to chromosome 22. And then this part of chromosome 22 will also break off and then attach to chromosome 9. This will give us 2 altered chromosomes, and the first chromosome here, it's called the translocation T922. So 9 is referring to chromosome 9 and 22 refers to chromosome 22, and this is going to be the first alteration. And the second chromosome will become known as the Philadelphia chromosome. And what happens in that chromosome is that the ABL1 gene, which is used to be on chromosome 9, is now fused with a BCR gene that is on chromosome 22. This fusion will result in this gene producing a protein that drives uncontrolled growth of white blood cells, and this uncontrolled growth leads eventually to myeloid leukemia. That's why this type of alteration it can be very fatal. The second type is the deletion, and in deletions, what happens is that a part of the chromosome gonna be lost, and an example of that is the Cri du Chat syndrome. And what happens in Cri du Chat syndrome is that part of chromosome 5 is deleted, and as we can see here, that this chromosome is shorter than its homologous chromosome, and that's because this part was deleted from chromosome 5. This deletion will result in having developmental delays, intellectual disability, also distinctive facial features, and this generally would show how changes in or abnormalities in chromosome can be very, very fatal, and also results in very severe complications. And here are 3 questions that are exam-like questions to test your understanding of today's tutorial. Hopefully you find them useful. And with that, guys, we reached the end of this session. Hopefully it was easy to follow and it benefited you. Also, please don't forget to like, comment, subscribe, and also turn on the notification button so you'll be notified of our future videos. Also, don't forget to check the time codes in the description. Thank you for listening.